Welcome everyone to another seminar. Today it is my great pleasure to introduce Dr. Mercy Akinyi. She received her PhD from Duke University. She also has a veterinary medicine degree from the University of Nairobi. And I promised her that I would keep my introduction short and sweet. So without further ado, welcome. Thank you.
Hey, great. Okay, I assume I'm audible to everyone. Cool. I think my initial remarks is just to say thank you to Anne and Sarah for such a warm welcome and for being my hosts. And for everyone else, it's good to meet a lot of the familiar faces. I was with at Duke, but also to meet new people and meet with a number of people here. It's been such a pleasant visit. And I'm also told the weather has been pleasant. So I'll try and think I brought the pleasant weather with me. But yeah, I've had a good time. And thank you so much. So today I'll spend time talking about some of the work I did for my PhD and some of the work that I'm currently doing. And I'll be happy to just have feedback from all of you. So on primates, behavior, and parasites. And this is the outline that I have, a little bit of introduction of where I am from, a background on disease ecology. Then I'll talk about the work I did on the drivers and cost of helminth infections in wild female baboons, a little bit about wildlife interaction and zoonosis surveillance, not so much because I don't have much time. And then I'll give highlights of other ongoing projects in my lab. So this is where I come from, Eastern Africa, from Kenya. And I'm based at the Institute of Primate Research, which is really a forest that is home to a few animals, but also home to a captive population of primates that are used for different kinds of studies. And this is in outskirts of Nairobi, which is the capital of Kenya. So a little bit of background. When we talk about disease ecology, it's really talking about the host and pathogen interactions in the context of the environments. And we do look at things like the transmission dynamics, as well as the potential effects of disease, whether they're beneficial or detrimental to hosts. And some of the terms that you'll hear me use a lot are disease risk or infection risk, which really means the probability of acquiring an infectious or non-infectious disease, but could also easily mean the ability to limit the establishment of a pathogen once it's in the body. And then one of the most interesting things in disease ecology is this, which is known as the epidemiological triad. And really it means for a disease to occur, there needs to be an interaction between the environment, the host, and the particular pathogen in question. So looking at these predictors of risk of infection or disease risk, if we look at the environment, it could be different things. It could be the social environment, such as social status, and I'll talk more about this when we're looking at the female baboons. But it could be what is external, like things like rainfall, biotic, in other words, rainfall, temperature, humidity, and habitat, all of which could affect the host directly or could affect the pathogen as well. And that's why we talk about the interactions between all of this. The host itself, it could be the age. The age will dictate whether a host will get a disease or not. It could be reproductive status for females. It could be whether they're male or female, and then genetics also. And of course, a lot of us know about gene-environment interactions. And also for pathogens, different things also will predict infection risks of hosts, such as the species, transmission, life cycle, and virulence. Things like the life cycle, really you're talking about, is it a direct life cycle where a pathogen will just infect a host, or is it indirect, meaning it needs a vector or an extra host before it can affect the definitive or the final host? And so all of these need to work. There needs to be interaction between these for the diseases to occur. So again, some other terms that I'll be talking about is these are actually the predictors of infection risk. But two main things do stand out. And this is one, host immune susceptibility and host exposure. So by host immune susceptibility, this is really the ability of a host to limit infections from progressing, or the ability of a host to limit itself from acquiring an infection, right? But host exposure really means the ability, well, not really the ability, the chance that the host will encounter infective stages of parasites. And normally these are the forces that drive infection, and they're not mutually exclusive, meaning they could be working together or they could be working separately, and this will produce different patterns of disease. So I'll talk about some of the work I did for my PhD on cause and drivers of helminth infection in baboons. And this work was done at Duke, but also in cooperation with Notre Dame University as well. So why helminths? There are very many helminths, but I'll definitely talk about nematodes. But helminths are ubiquitous, meaning you see them all over the environment since time immemorial. And a lot of that, they could be generalists, meaning they will infect a wide range of hosts, or they could be specialists as well. And 43% of pathogens in primates are helminths, and some of these are actually zoonotic. And helminths are associated with either direct costs or indirect costs. By direct costs, what I mean is that they cause pathology to the host, like they will, they could form nodules in the intestines or just disrupt the intestinal mucosa. That would affect how hosts are able to absorb nutrients, and this will have downstream effects on the host body condition. Like I think working as a veterinarian, like you can see a particular cow and you can know that it has helminths because of how its fur looks like and how thin the body is. So those are direct costs that you can be able to quickly pick up. But there are also indirect costs, and these are thought to be hosts will end up spending a lot of time and energetic resources fighting helminths, and so they will not be able to have resources for other activities such as reproduction, lactation, things like that. So there's kind of like a trade-off of some sort. So those are the main classes that we can have of the costs. So going back again to this picture that had the epidemiological triad and just seeing what we could see in primates, a lot of this remain the same, but I think I said I'll talk about social status and group size. And primates live in different in different sizes. So you'll find some groups have many individuals, some groups have fewer individuals. But also one big thing is the social status, and this has to do with ranking or what is called dominance hierarchies. And for some of the primates, like specifically for baboons, you'll find male and female hierarchies are kind of established differently. Like males will fight to be at the top, and females will kind of ease their way up, mostly because it's matrilineal. It depends. If your mother was high ranking, then you're likely to be high ranking. So all of those contribute to what we term like a social environment. So again, coming back to this, let's keep in mind that these predictors will still work through the same way. It's either immune susceptibility or host exposure, and they are not really mutually exclusive. So I'll give a few examples of hypotheses or predictions. So for example, if you pick group size, if you look at hosts susceptibility, the thought or the assumption is the more individuals are in a group, the more they'll fight for resources, and so they will not get what is probably optimal resources. They will get what probably is like the bare minimum. But if a group is smaller, everyone is likely to get like an equal amount of resources, and no one is fighting in terms of the dominant individuals will get more resources than the others. Everyone has a way to get the food. And so you will expect that the helminth burden will have a positive correlation with group size. As group size goes up, the helminth burden goes up. If you look at host exposure, If you're in a group that has many people, it's likely that there'll be contamination of the environment that you stay. And that means there'll be more parasite infectious stages. And that means that hosts are likely to get exposed. And so the direction for both susceptibility and exposure will be more of a positive association. But let's look at ranking. So if you look at host susceptibility, the higher ranking animals have access to resources, are well connected, they're in good body condition. So the assumption is that they're able to fight off infections. However, the low ranking animals, it's the exact opposite. So we'll expect this positive correlation. But when you look at exposure, we kind of see a different kind of correlation. The assumption is that the high ranking animals probably have more access to food, and they will probably pick up parasites faster compared to the low ranking ones. Then we have one last one, rainfall and also temperature. This could happen. And hosts will thrive well when they're optimal levels of rainfall. If there's too little rainfall, host body condition is not good. If there's too much rainfall, there's probably also not enough food in the environment. So there needs to be some kind of optimum, right? And then for exposure, what happens is too little rainfall or too much rainfall also will affect parasite infectious stages. Too little, the parasite stages will die off. Too much rainfall, the parasite infectious stages will be wiped off. So there needs to also be an optimum of some sort. So that said, the study that we did, we tested how host group and population level processes predicted female infection risk to helminths, and then also tried looking at the costs of helminth infection. And the main costs we were looking at were female interbirth intervals and stress hormones. The interbirth intervals, we thought about this because there are studies that have shown that if you remove ectoparasites from particular animals, you increase their reproductive success. And then there was also a study in one of the primates, I think in geladas, where helminths were associated with prolonged interbirth intervals. And of course, if the interbirth intervals are prolonged, then it kind of means that you're kind of lowering fitness, because then you can't produce offspring as fast or return to cycling as fast. And this work was done in Amboseli, which is in Kenya. And these are yellow baboons, though in this population, they're not purely yellow. There's a lot of admixture with what are known as the olive baboons. And this is a study site or study project that has been there for the last 5 decades with extremely good and detailed data. And they do monitor the animals. There's plenty of data for this particular population. So I feel like it was good to be involved with this kind of a project. So let's go back, moving away from the triad and just explaining how we kind of put our predictors of infection risk. So there are predictors that we felt were working at the population level. And this really means everyone in the population is experiencing it. So for example, rainfall and temperatures, when that's hot, everyone is experiencing it. And then there are what we call the group level processes. And this was group size. And so different groups would have different predictors working against them, depending on how big they were or how small they are. Then there are those that work at the individual host, which really is for each individual, what's going on for that individual, the age of the individual, the social status, is the host highly connected? And connectedness for us in this case was more of, it was constructed from a grooming data because primates really groom a lot. And it showed kind of like you have people who care about you. And social connectedness had been shown to have many benefits, inclusive of survival. And then there's also an aspect of co-infection, whether you're infected with other parasites or not. So these are the different, this is how we tiered our experiment. So generally, what we did is collected 745 samples over 5 years from 122 female baboons. Very painful to do microscopy for all of these samples. And some of this data include the host environment, as we had talked about, the social status, the social connectedness, hormone levels, and reproductive indices. And for this population, it's easy to track what stage the animal is during reproduction because they normally have sexual swellings. So as I mentioned, fecal parasitology using microscopy, which is flotation and sedimentation. And really what you see is you try and look at the eggs, try and look at the larvae, and then you have like an atlas and you're able to know which species is infecting. And then you go through the painful process of counting the number of eggs for at least 7 slides per animal. And then we also would measure the parasite burden by presence absence, meaning whether the parasite was present or not. And then parasite richness, which is really the distinct parasite types that were present in the population. Fecal hormone analysis is also some of the work that we spent time doing. And it's collecting samples in ethanol and storing them, taking them through freeze drying. So this is done in the field. This is done at Nairobi University, which is a partner of the Baboon Project. And then the rest of this was done at Duke, some of it at Duke and some at Princeton. And it's methanol extraction and then conducting radioimmunoassays to figure out different hormones. So for our case here, it was estrogen. Well, we didn't do estrogen, but they normally do estrogen and progesterones and testosterone, but we were more keen on glucocorticoids because that is the signature for stress. Then also data mining, and this is just, they have a fantastic database that then you can get all the demographic data from the age of the animal and anything else that the animal has experienced and the behavior and the ranks over time and even grooming. Because for each of these measures we were taking, we had to look back. So for example, for rainfall or for grooming, we took like 3 months cumulative data to find out how much grooming was received 3 months prior to collection of these samples, how much rainfall was there 3 months prior, because it kind of gave a good index. And so having a good database is good because you can retrieve all of this information. So the data analysis was a lot of linear mixed models, and really we had different variables. So as I mentioned, whether it was presence absence or parasite richness or the actual helminth burden, and these are fixed effects, which we had already talked about previously. Oh, and we had the random variables because for some of the females, we got several samples from them, not just one. So these are some of the helminth species that we found, the most prevalent one being Trichuris trichura, but then you can see we have a whole other list of helminths here. Strongyles, as much as their eggs look alike, they do form different kind of species. So like these are different genuses and species. So you really cannot be able to say that the strongyles is either one of this. It could be either one. And then we have parasites that undergo indirect transmission. And direct transmission is like fecal oral, like the infectious stages are in the environment and the host can pick them and get infected. Indirect means that there needs to be a vector in between. And the vectors sometimes are normally dung beetles. So the A. caucasia and S. pigmentatus both undergo indirect transmission. Of great interest, especially for me as a veterinarian, are these ones that have stars, because they're zoonotic helminths. This means they can be transmitted between humans and animals. And parasite richness, meaning unique parasites, there were quite a bit, 0 to 5 species with a medium of 2 species. So just picking one of these, and this is for Trichuris burdens, and seeing the different profiles, we can see the age. So infection parasite burden increasing with age, and this is probably a function of immunosenescence, meaning the immune system is getting old, and so the animals cannot handle, or it could be cumulative, meaning the animal has actually picked parasites as it's growing. Then we have co-infections. So if you have strongyl infections, a higher probability of you having Trichuris, and that could be because their route of transmission is the same, and so they will co-occur. And then for social connectedness, not super strong, but there is a trend where you'll see the animals that are isolated which is up here, they have more parasite burdens than those that are well connected. And then group size, as the group size goes up, parasite burden goes up. And then for both rainfall and temperature, parasite burden goes down. So they have negative associations, meaning most likely the high temperatures will kill the parasite infectious stages, and high rainfall will probably wash away the infectious stages. So I think I don't expect us to go through the whole table, but this is just to point out that you have different parasites, right? This is like different parasite measures, but you can see for each parasite, they have different effects that contribute to their burden. So it's not a one size fits all for all the parasites. And then we have interesting things like, for example, social connectedness here. As connectedness is increased, the trichuris load is decreased. But if you look for a different kind of transmitted parasite, like Caucasia, for here, as social connectedness is increasing, then there's a positive correlation. And this one is indirectly transmitted. So it's, again, it's not a one size fits all for social connectedness. Again, for parasite richness, which is an overall measure, as you increase the connectedness, the richness reduces. So a lot of different ways to think about what's going on, and so many things that could possibly be modulating the burdens. So when we looked at the costs of infection, a good thing to see is interbath intervals. This is just the general measure between successive baths, and especially for females that already had children who survived up to 1 year. And you can tell, if you look at all of these indices, you can tell that as measures of parasite burden increase, so like that's richness going up, trichuris going up, S. pigmentatus, strongyles, and strongyles, these measures of interbath or components of interbath interval, all of them are long. So they are prolonged. So that means the parasites are having an effect with the interbath intervals. They're prolonging this, and probably that's a cost for infection. For the glucocorticoids, will not claim causality, but when we do put models, we see that for glucocorticoids versus T. trichuris egg counts, there's some kind of a positive correlation. And this could possibly mean the energetic costs that the hosts are using to fight off infections are extremely high. So the conclusion for this study was really a comprehensive investigation of the drivers of the helminth burden, and there's multiple levels influencing the burdens. And we see both components of susceptibility and exposure, though these are difficult to entangle, and definitely their detrimental effects on hosts, especially with regard to fertility and stress, which I've already mentioned. So I'll move on to, again, some kind of work that we did, I think a few years ago, and that I'm currently doing for some of the projects. And this is human-wildlife interaction and zoonosis surveillance. And most of this work will just talk about what we did. So this particular study is based on a lot of what is going on right now with regards to One Health. And the thought is humans really impose a lot of anthropogenic pressure. So these are activities that destroy the environment, such as deforestation and eating bushmeat, but also climate change, amongst other things, illegal animal trade and intensive animal farms. All of these are factors that actually affect the ecosystem. And you know that wildlife are within this ecosystem together with zoonotic outbreaks. And so these influences, especially when they're negative, they're likely to bring about the rise of zoonosis, and this leads to a spillover. So SARS-CoV is one of the things that has always been believed to be a spillover of events. But the most interesting thing about these aspects of spillovers, and when there's a spillover, it will affect domestic animals, pets, wildlife, humans, but nobody ever thinks about this blue light line. What about the reverse spillover? I know some of the clear cases, again, like SARS-CoV, when we had, so SARS-CoV was thought to have originated from pangolins and bats, and then it spilled over to the human population. But from the humans, it came back to the animals, right? There were deers that were affected, chimps, cats, dogs. And so a lot of times we as humans spend a lot of time thinking about spillovers, but nobody really focuses about the reverse spillover. So we tend to believe that a lot of the work that we do for surveillance, for primates and for other animals addresses both spillover events and reverse spillover events. And this provides room for prediction, detection and control of emerging infectious diseases. So for this particular study, we wanted to determine the risk factors associated with transmission of zoonosis at the non-human primate-human interface, so areas where there's interaction, and then to determine the prevalence of viral and parasitic zoonoses. And lastly, to engage both stakeholders and communities about how they can control transmission within this particular area. So again, I won't go into details about the methods we used because we don't have so much time, but what you can see from this particular population is a lot of human activities. First, there's a lot of poverty, which will drive the human wildlife interaction, so the social economic status. And you'll see that a lot of the livestock that is there, they share resources with the humans. So where they go and drink the water, that water is used by both livestock and wildlife and humans. So if there's anything that is being transmitted with regards to water, then you expect for that to happen. Then a lot of deforestation and logging. This is charcoal that has been retrieved from one of the forests, which happens to be a habitat for the animals as well. And so this just shows some of the risk factors that are there. Other risk factors include eating bushmeat. So this was one of the respondent houses that we went to, and they were like, yep, I do get bushmeat and I'll dry it up and I'll eat it over time. And so again, the way the meat is handled and how it's kept, no one is really sure about it. But looking at here, when you're looking at animals being hunted for bushmeat, a lot of the people eat antelopes, but you can see here that they will also eat monkeys, right? So that says a lot because for most of the diseases that we see that are emerging, it's normally about contact. It's not so much about how well do you cook the meat. It's like, how are you handling the meat right from the beginning and the fluids that are coming from it. So this was interesting to see this whole array of animals that are being eaten for bushmeat. But when we narrow down to the monkeys, because the work was focusing on primates, you can see it's different things, right from hunger, and hunger really topping it up, because like these are environments that are semi-arid, so they don't have constant water. And so food is a big issue. And then it was just because there's a belief that monkeys will taste better than others for curiosity, others for revenge because of crop raiding. And then there were also cultural thoughts. There are people who feel that if you eat a monkey, it's supposed to help you in one way or a particular way. So again, cultural thoughts were coming in here. And just to show you some of the level of conflict. So these are common species in IPR, baboons, vervets, and sykes. And baboons are the most dreaded because they're the ones that participate in livestock predation and also in crop raiding. Yeah. And what people will do is, even once the baboons have attacked the livestock or the crops, whatever is left over, they will still use it for their meals and other things, which then forms a way for transmission of pathogens. So when we look at the different zoonotic diseases we mainly worked in two sites, Tana River and Savo. What you can see is these are kind of the zoonotic helminths I talked about. You might not see the names clearly here, but these are different zoonotic helminths that we did find during this study. And what was more exciting is we did go to clinics. So see, this is Washington County government. So we went to the clinics and we took their books, and because they're not digitalized, and we went through each of these books just to see which are the common parasites that they report in the clinic, and how does this compare to what we were seeing. I think that was because with our study, we didn't have enough money to conduct human surveillance as well. But a typical One Health study would require that you look at the animals, domestic animals, wild animals, the environment, and then humans as well. So this was a way of, I won't call it a shortcut, but a way of at least overcoming the particular bit of not having funds. So for this particular one, what you can see, this is Entomoeba, which is a protozoa. And this was common in non-human primates, which is red, but also common in humans. And this is not surprising because of different habits in terms of people's defecating mechanisms, like they're not latrines, so they will defecate anywhere. Some of that will get into the water or into the streams that are being shared by both animals and humans. The other one is here, Schistosoma mansoni, and this really is normally, you get it from water, yeah, and from snails. And then also hookworms, though not as many in humans. And then the last two, not as much in humans, but you can see that there are parasites that are being shared by both the primates and the humans. So we did look at prevalence of non-human primate viruses, and of importance are these two, foamy-viruses and chikungunya. The herpes one, probably important for the primate, but not so much transmissible to humans. The foamy and the chikungunya, these ones are kind of dangerous because, and this was more of just showing exposure, but they're dangerous because it means the primates then are acting as reservoirs or sentinels, which means mosquitoes can come and bite them and take the infections back to humans. Oh, sorry. So the other extremely important part of the work we were doing is once we gathered all the data from the clinic, from the animals, we went back to the communities because we thought it was important for us to tell them, this is what we found in the community, and these are the risk factors that can make you get zoonoses, and this is how to mitigate them. And also allow the communities to give us feedback in terms of why are they doing the things that they do, right? But we also had a separate stakeholder workshop where we also gave them feedback from the community and told them, look, this is what the community is doing. What are you able to do? Are you able to create more community projects so that people, you can elevate the social, are you able to do like frequent outreach programs so that you can teach the community about the zoonoses? So it was good to have a feel from both the community and the stakeholders. And so I'll just bounce through the remainder of the talk and talk about some of the things that we are now doing and some of the main focus of my work, which is a lot of it now is zoonotic surveillance. And during COVID, I got a grant that allowed us to look at coronaviruses in understudied primate populations. These are colobus monkeys. This one is the red colobus, which is critically endangered. This is the colobus gureza, and these are cyc monkeys, which most people don't study, from different sites, as you can see. And what we're doing is just screening to find out if they have any form of coronaviruses. And we did see that they don't have SARS-CoV, but they have some of them have the endemic coronaviruses known to humans. So that is still work that we are analyzing. And during that time, we also got a chance to provide support to the national government with regards to surveillance. And I think Joseph will be here 2 weeks from now. He was heading this particular initiative. And at the time, I also got a chance to learn how to do genomic surveillance. And because we are still a small institute and we cannot afford many things like the Illumina sequencing amongst others, we got through this grant, this nanopore, cute nanopore device, and we tried it out and it was able to work. And then once we tried it out and got some data, we didn't know what to do with the data. And so that's where Anne comes in, right? So Anne and Rebecca spent some time with us in Kenya, and they just took us through this bioinformatics training and pipelines of how do you analyze genomic data, yeah? And so as to provide a little bit of capacity. So this is still new for us. It's something that we still hope to expand. Instead of just being focused on genetic analysis, we want to go deeper and delve deeper into genomics as well with regards to pathogen surveillance. Then one of the ongoing projects that we currently have is surveillance of zoonotic tick-borne diseases. These are transmitted via ticks, and these are some of them: Babesia, Rickettsia, Borrelia, and Anaplasma. Again, these are able to get to humans as well, but nobody pays a lot of attention to these particular parasites. The other thing that one of my master's students is doing as well, instead of just going under the microscope and looking to see which eggs exist, we are trying to also do molecular characterization. And after a very good talk with, I think it's called Arvin, it was nice to know that we can even do much more than just characterizing this. We are able to get much more data from stool samples. So we hope we can expand this. Then one of the PhD students, in collaboration with Notre Dame, is looking at the members of the gut microbiome for baboons and livestock in the Amboseli population. Will probably add humans as well. And what we are looking at is protozoa and fungi as well. And she's already gotten some data that indicates some zoonotic protozoa and fungi. So this is very interesting for us, because now we are starting to have this kind of a One Health sampling. Instead of just doing primates only, we're starting to extend to other components of the ecosystem. And then one of the work that I'm still winding up on was my postdoc work, which focused on a lot of malaria genetics and drug resistance, but also co-infections. And this one is just looking at proteomic profiles of baboons co-infected with multiple parasites. Yeah, and with this particular one, what we looked at is we saw that parasites that have schistosoma are protected from Plasmodium. So we wanted to know what are the mechanisms behind it. So that is still in the write-up. And those are the different funders and different supporters of the work that we've been doing over the years. And I'll be happy to take questions.
I will run around with questions.
So I was wondering if you could talk a little bit more about that Plasmodium schistosoma association. Do you know what the mechanism is yet?
That from the proteomic profiles that we did, it just seems like what drives the Plasmodium infection has to do with a lot of like what is called metabolic crisis. That's what the proteomic profile shows. But the assumption is whatever the worms are producing are protective, and so it helps the animals not undergo that crisis. That's from the proteomic profile. But there's like 2 papers that have been published by one of our colleagues, and she talks about the innate immune system being the driving force of that particular mechanism. So I can refer you to those papers. I can send them to Anne. Yeah.
Hi, Mercy. Hey. Thank you so much. That was an awesome talk. I asked you a bunch of questions yesterday, so I have fewer today, but I still have a bunch. So for the Amboseli paper, do you think about things like infection intensity and species richness as kind of fairly stable traits of individuals? Do you expect them to remain static for those females over time? Or are those things, like if it's like related to rank, does it end up being kind of stable trait of a female? Or do you expect it to vary kind of randomly or along with other variables?
Yeah, I think that's a good question. I would think it would vary, and it would vary because of many things, even seasonal effects, as you can see with rainfall and temperature. It will not be stable, and even probably with the rank as well, especially for males. Rank is never stable, and there's a parallel work that was done for males. So I think my answer is it would be good to do like probably follow those females for a while and be able to come up with that kind of data. Yeah.
Cool.
Yeah.
And did you say you had 7 slides per fecal sample?
Yes.
Oh my God.
Yes, because because for 2 samples you have to do the flotation. At least the flotation is good because there are particular eggs that will float up, and then there are particular eggs that will sediment. And so if you do flotation, you cannot find them. And then the ones for sediment, if you do, the vice versa, you won't see them. And the flotation is easy and cleaner. So that is fasciites too. But the sediment is a nightmare because you have all sorts of things, soil, mites, and all of that. But like the most beautiful of eggs are actually, you'll find them in the sediment. So I don't know, but it was painful. And one of the things that I was gonna talk to Noah about is having like artificial intelligence to be able to look through these slides. So we stopped counting eggs and just moved to richness because it was just too painful. That's all I can say for 7 slides. Yeah. So that's how we ended up with 7 slides. So 5 for sediment and then 2 for float. Yeah.
Okay. Awesome.
Yeah.
And then last question. For the COVID study, were you sampling populations that you expected to have some sort of exposure to humans? Or how did you select those populations? And have you thought about retesting now that it's been some time to allow for some more interaction?
Yeah. So for the COVID testing, it was a mix of both. So for the colobus, especially because we really cannot track them, we just got stool samples and looked at that. But it was during COVID. And then for the psych monkeys, some of the samples were before COVID, some were after COVID. So we didn't see much. And I don't know about doing a follow-up study if we will really get much, especially from what we saw. But the Sykes monkeys have more interaction with the humans. The colobus are really arboreal and they're up high. So maybe that's why we didn't see anything in the colobus monkeys. But we also did what is called the ACE2 receptor diversity to see how similar their ACE2 receptor. So the ACE2 receptor is the one that allows COVID to infiltrate hosts. And when we looked at that across all the species, it was all similar. So the assumption is all of them should be able to be predisposed. But more interestingly, where I work at the Primate Center, we had a lot of cases of COVID, but these primates never ever got COVID. Part of it is because we were always in masks, so maybe that protected them, but they're also in outdoor facilities, so maybe that also protected them. But I don't know if we can mount another study. Yeah, the permitting is too chaotic to do, but unless it is done non-invasively, which we still are doing, we're still collecting fecal samples. Yeah.
Cool. All right, I will cede the microphone.
Thanks for your talk. That was really, really fascinating. I was interested in your study where you look through the books of like human cases, and I'm sure you know that you're missing stuff, right? If you do it that way. So can you talk a little bit more about that? Are physicians routinely testing for stool samples for any infections, or is that really if there is a very specific morbidity that they need to test it for?
Yeah, I think a lot of that is whoever needs to come to hospital, like the very sick patients. Those are the only places you'll be able to detect the parasites. But it's likely that a whole lot of the community has the parasites. And so the best approach, I think Joseph was here in 2 weeks. He's been able to do multiple species at the same time. So he's been able to sample the humans, primates, some livestock, and rodents, and bats as well at the same time. And that definitely gives a better picture. But for us, we thought, well, given that we sampled animals during this time and we are still in this facility, it would be nice to just see what is commonly reported. And stool, the beauty about helminths is that microscopy is very easy to do. And so a lot of the facilities have the capability to do it. But you're right, it's not probably as accurate, but it does give some signal, which I think is good. Yeah.
Any other questions?
Thank you. Okay. So, for the foamy virus and the chikungunya, are those, you said they could be reservoirs, the primates could be reservoirs, and then it could be vector-borne transmission. Are those also things that you might anticipate could be directly transmitted during hunting or meat dressing?
No. Those are all vector mediated. And actually from those particular areas, humans also get, especially for chikungunya. For foamy, I haven't seen a lot of work for foamy, but for humans, yes. There's quite a bit of chikungunya, yeah. Mosquito transmitted.
So it looked like there were the parasites that are the helminths that are zoonotic that would be transmitted through kind of like the water soil transmission route. What would you expect to see, or like what did you say to people about like, you know, being careful during meat dressing and hunting and that kind of thing? What could be transmitted?
First of all, the people asked us if we were from the government, and we said yes. but we don't care what we're doing. We just need data. And they were happy then to open up and tell you what they were doing, because when they see a blue-plated vehicle, that's a government vehicle, and they don't want to talk. But we definitely told them that it is wrong to eat the bushmeat. Under no circumstance should they eat the bushmeat. But we also talked about basic things like hygiene, washing, disinfecting, their water that they use. So those are just like the basic things that they could potentially do. And then throwing out the crops that have already been raided instead of trying to still use them for their own meals, to probably just throw them, which we are sure some of them will not do it. So yeah, and also asking them ways that they think they can help solve the problem. Yeah. Yeah. Thank you.
Okay, any last questions?
This is more of a curiosity based on one of your slides, but so one of the reasons that people eat the primates is because they predate livestock. And I was just curious what kind of livestock they predate, because I can't imagine them eating a cow.
Yeah, yeah, that's true. I think a few years ago, when was this? Maybe 2012, 2013, I can't remember. We had this, you know, the male baboons really have sharp canines. And so, you know, like goats, the babies of goats, kids, they will eat those very quickly. But they also actually will like kind of like try and also predate on the bigger goats, though not successful. So I spend a lot of time trying to treat goats because the community were upset and they killed quite a number of baboons at that time. So I had to go out and talk to the community, tell them, let me see the goats, let me see the kids. And then, yeah, so that I've definitely seen. But yeah, the smaller, and chicken, things like those are smaller. Yeah.
Thanks, Mercy. So just hearing this made me think, do you have a sense for whether there is local knowledge about zoonotic transmission, kind of like, you know, indigenous knowledge in which they might know that certain kinds of things, like if crops are raided, that might be have to be processed in a different way. Certain meats have to be boiled, not just roasted. Do you get any sense for those kinds of things?
Yeah, there was that. They're like respond, and this is in Danson's paper, actually. This is a lot of Danson's work. We worked with him. And there are people who are like, you cannot eat the monkey. It's a taboo. Don't go anywhere near it. And then there are those who are like, based on experience, they ate the meat and they had a running stomach, so they've never gone back to doing it again. So there are pockets of people who understand what's going. Then there's the pockets of people who don't care, and they'll just do what they want. Yeah. So I think the outreach is important. We're just finishing from a different site. We're finishing the helminth and protozoal work, and we still are going to go to the community and also talk to them and hear what's going on with them as well. Yeah.
Hey, any last question? All right, then I would like to once again thank Mercy for her wonderful talk.
Thank you. Thank you. Okay.